start up. Perfect. All right. Hello, everyone, and thank you so much for coming to attend our third Trainee Research Presentation series of 2021 for the Women's Health Research Cluster. This is always a great way to learn about exciting things that other trainees in the cluster and from outside the cluster, what they are doing. And they're also a great way to practice talks if you have other talks coming up, things of the sort. At the end of these presentations, I'll talk to you all about when the upcoming trainee presentation series is, if you feel like you would love to present after seeing these wonderful presenters. So to start things off, I would like to first acknowledge that we live, work, and play and participate in a community on the unceded, ceded, and traditional territories of the 203 First Nations, along with 38 Métis chartered communities, each of which possesses their own unique traditions and history here on this land that we now refer to as British Columbia, which is where I am presenting from. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's Call to Action, the United Nations Declaration on the Rights of Indigenous Peoples, and the BC Declaration on the Rights of Indigenous Peoples Act. In all our work, we are committed to ensuring Indigenous women's rights to health and safety, and the equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. So today, as I was saying, I'm joining you from Vancouver, which is part of the unceded homelands of the Coast Salish peoples and the traditional territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. I also wanted to add that in case you are wondering if places to donate for Indigenous peoples and Indigenous women's health, I will be sharing a couple links in chat to some international organizations as well as Canadian organizations at the near the end of this presentation while making a few other announcements. And also it is the National Day of Truth and Reconciliation for Canada tomorrow. So I will place a link for more information about that in the chat also after the presentations. Also, I would like to acknowledge our other sponsors, including CIHR, Michael Smith Foundation, Biotalent Canada, as well as UBC. And if you like what you hear, I highly suggest that you tweet about it as the presentations are going on or after the presentations, and feel free to tweet with hashtag #TraineeBlitz and tag us on that. So yes, we will get started. And just to let everyone know who is joining us, the presentations will be about 5 minutes each, and there will be about 3 minutes for questions after each presentation. So let me stop sharing right now. And to start us off, we will have Jodie Hughes. Jodie is studying disparities in the care of people with endometriosis and identifying where there may be gaps. Jodie will be collecting health-related quality of life scores using an online survey and comparing the results between sample groups. There will be semi-structured interviews with people that have expressed an interest in getting involved to add qualitative weight to quantitative results. And today, Jodie will be giving us a presentation titled Study on the Effects of Location, Ethnicity, Gender, and Care Approach on People with Endometriosis. So awesome. Jodie, feel free to start sharing your screen.
Thank you. And we are going to... Is that all up yet? Brilliant. Right, so as our lovely Travis said, I'm Jodie Hughes, and my PhD study is looking at endometriosis care and how it differs between sample groups of people. So endometriosis is a chronic whole body condition where cells that behave in a similar fashion to the endometrium are found outside of the uterus. All the treatments for endometriosis can negatively affect patients' quality of life. This is like what my research is focusing on. So it affects 176 million people worldwide. The diagnosis time on average is 7.5 years in the UK, and that figure hasn't changed a lot in the last 10 years. The condition costs the UK economy £8.2 billion a year, and in the US, the average cost is around $30,000 per patient per year. This all has a massive effect on the patient's health-related quality of life. Part of the reason that endometriosis isn't diagnosed sooner is due to the normalisation of period pain and symptoms that are associated to menstruation. If a young person's not learning the signs of menstrual disorders, they're led to believe that the symptoms are normal. So past research into endo has often left out key demographics of people, including people of colour, different socioeconomic backgrounds, and different genders. The research that has taken place has often been centred on the late 30s white career woman who historically got a diagnosis through chance when undergoing investigations for infertility. Studies show that people of Asian ethnicity are 9 times more likely to develop the disease, but no one knows why. People of Black origin are more likely to be dismissed when talking about their pain with a doctor. This makes diagnosis times longer and makes it harder to access appropriate care. Transgender men are often excluded from endometriosis studies, but this demographic of people is more likely to be medically gaslit by professionals that are meant to be treating them. And women past their menopause or that have had a hysterectomy are excluded from many endo studies because there's a misconception that endo goes away once the body has gone through hormonal changes, which isn't the case. My project is mixed method study using online survey to collect demographic data and health-related quality of life scores using the endometriosis health profile that was developed by the University of Oxford. As well as the pelvic pain impact questionnaire. These quantify the impact that the symptoms of endometriosis has on mental and physical health of patients. So the higher the scores, the more the impact that's had on the lives. As Travis said, I'll also be doing semi-structured interviews with participants and they will be coded using interpretive phenomenological analysis to identify common themes. Preliminary results from the survey. So I've just reached halfway. We've had 900 people take part now. So preliminary results are showing from the survey show us that when we look at the mean percentage of scores from the endo health profile questionnaire, you can see that it's about the mean is around 40%. And the mean for the total scores on the pelvic pain questionnaire are around 13 out of 20. So the higher scores are telling us that the overall health-related quality of life of the people that are engaging in the survey are low. Preliminary results also show us that more than 70% of people that took part in the survey reported interruptions to their care due to COVID, which isn't surprising, especially here in the UK, because the gynecological units were the first to be redeployed to the COVID wards and were the last to go back to their normal jobs. Almost 40% of the people that filled out the survey reported that they felt victimised because of their gender when seeking care for endometriosis. The quotes that you can see have been taken from the couple of qualitative interviews I've done so far. They're talking about interactions that have happened within the last 12 months. So telling a patient to drink whiskey when she's experiencing pain during intercourse shouldn't still be a thing in 2021. There should not still be a racial bias when you walk into the doctor's office to seek help for symptoms of endo. This study will identify gaps in knowledge and care and research in endometriosis. But with these gaps identified, the data can be used to inform policy on how care and treatment can be changed to improve the health-related quality of life of patients. And thank you for having me. It's been great to share my research with you and look forward to answering any questions that you may have.
Awesome. Thank you so much. And yes, I love how everyone already does the electronic claps. Feel free to do electronic claps for Jodie.
Oh, thank you.
Love that. If anyone has any questions, feel free to either type them out in chat and I can read them, or to just unmute yourself and ask some questions too.
See, it must have been that good that no one has any questions about it.
It was a really good presentation.
Oh, thank you.
So thank you so much. A question that I can ask, just seeing some of those, the couple comments that you did put up there as examples, I'm wondering, what were some of the most common comments so far in the day that you think that you have seen, and maybe some of also the most, well, I think those were kind of, well, some of that are more extreme comments as well.
Yeah. So the most common is normally after people have been diagnosed is saying about validation, is how once they had that diagnosis, fighting for between 10 and 15 and 20 years sometimes. After that, they feel really validated, which is awful because they've been made to feel for that long that they're insane. I spoke to one person, and to get a diagnosis of endo, you have to have an operation. You have to have an investigative laparoscopy. She had 2 of those, and they were telling her that she didn't have endo. And then she paid to go private over here. And they found stage 4 endo, like she had to have part of her bowel taken away, and she had to have a womb separated from her pelvic wall. And it's like, why is this still happening? Like, we first diagnosed endo in 1860. Like, why are we still hearing the same things now that we heard back then? Like, yeah, that was the most common thing, was like, it was so relieving when they finally turned around and said, yeah, you have endo. But there's just been so many horror stories, so many. And the whiskey one was, that just blew my mind. It's why I had to put it into the presentation, because it just blew my mind that a doctor last year told somebody that they had to drink whiskey before they had sex because she was just too frigid. And it's like, oh my God, why is this still happening?
Mm-hmm. And but I can't remember, I just think I've forgotten, but where's your sample coming from? All of your samples?
Everywhere, all over the world. Yeah, we've just hit the 1,000 mark and hoping to still get a few more. So yeah, it's all over the world.
Yes, I can be very interested to look at how those different areas,
definitely
commonalities, to see how we could change like separately based on each area. Stephanie has a question. I will see if I can say it correctly. Here we go. This may be a bit outside the scope of your immediate research, but do you know if there are biomarkers or anything being developed to make diagnosis or early detection easier? Great question.
I wish I could say yes. I really do wish I could say yes. I know that in Edinburgh, there's a scientist, Professor Andrew Horne, and his team, I think, have just maybe identified a biomarker that could potentially be linked to endo development. So yeah, it's definitely worth keeping an eye on the stuff coming out of Edinburgh, because that seems to be amazing at the moment.
Awesome. And Tanvi, you unmuted. Did you have a question? No, maybe not.
Nope, just accidentally unmuted.
Okay, sorry. No problem.
But great talk, Jodie. Thank you.
Thank you.
Jodie. Amazing talk. Another round of electronic claps, please, would be great.
Thank you.
All right, perfect. So we will move on to our next speaker now. Our next speaker is Shilpa Rao. Shilpa is a PhD candidate in molecular medicine, PhD program at Cleveland Clinic and Case Western Reserve University in Cleveland, Ohio. She is currently studying the role women's lifestyle may play in the risk and progression of Parkinson's disease in Dr. Ignacio Mata's lab at Cleveland Clinic Lerner Research Institute. Shilpa completed her bachelor's in molecular biology at Loyola University, Chicago. And today Shilpa will be giving a presentation titled Constructing a Integration of Lifestyle, Clinical, and Genetic Factors in a Novel Parkinson's Disease Prediction Tool for Women. So take it away, Shilpa.
Yeah, sorry, I changed the title
No
before,
problem.
so.
No problem.
So as you guys know, this is my PhD project, and I'm just going to jump right in. Parkinson's disease is a neurodegenerative disease in which dopaminergic neurons are degraded in the substantia nigra, and this causes an aggregation of alpha-synuclein proteins and accumulation of Lewy bodies. And as I said, Parkinson's disease is a multifactorial disease. And what this means is that environmental, lifestyle, and genetics all play a role in the risk and progression of Parkinson's disease. So not only is it a multifactorial disease, but it is also the second most common neurodegenerative disorder of aging. What we also see with Parkinson's disease is that it's heavily sexually dimorphic. We see that men are 1.5 times more likely to have Parkinson's disease than women. But we also see that the symptoms between men and women, from motor symptoms, non-motor, and other measures of disease severity, such as health-related quality of life, are very different between men and women. So our lab is interested in better understanding the minority group of Parkinson's disease, and we want to look at women because care for women with PD are often overlooked clinically. And we see this through diagnosis and treatment outcomes. We see from diagnosis, women are often more misdiagnosed or undiagnosed than men. And this is because they have onset depression and anxiety, which is misdiagnosed as just something else other than Parkinson's, which causes their clinical manifestations to grow somewhat different, treatments and care to be very different because they're later stage and more severe. And the treatment outcomes are more severe in that they have a lower health related quality of life. So our lab wants to see if we can integrate lifestyle, clinical, and genetic factors to improve prediction and care of women with PD. And so we hypothesized that by creating a prediction model that combines both lifestyle, clinical, and genetic factors, we can better predict the risk, progression, severity of PD compared to the current predictions models in the field, which only look at either clinical or genetic factors. And how we did this, and it's a great leadway from Jodie, is that we are also starting, we made a survey, and that is women specific and their association to Parkinson's disease. So I just took a quick video. I'm sorry for the little, this combobulated words, but basically this survey goes through when their Parkinson's started from menstruation to menopause and how this affected their, did menstruation affect their symptoms? Did menopause affect their symptoms? And did they notice anything else about their cycles in relation to Parkinson's? And this is just a constant repetitive list of the symptoms that we're interested in seeing if they got better or worse in relation to these questions. We also wanted to see if there was any changes for those who had Parkinson's during perimenopause. And we wanted to see if they had to change their medication in order to alleviate those pains. We also ask about differences in surgeries, hysterectomies to oophorectomies, and if this affected their symptoms as well. Hormone replacement therapy is really big in the field, and we wanted to better understand that in women. And so a lot of papers don't look at the symptoms and how it changes and the different types of hormone replacement therapy PD women go through. We also ask about contraceptives, as that's a very contradictory field in the PD world. And we also want to ask more about pregnancies. Did they have trouble conceiving? Did they go through IVF? Did they have any peri or postpartum issues going with having PD? Lastly, we wanted to ask about different types of syndromes. Did they have breast cancer? Did they have PCOS? When did this start? And so this was just kind of a template because there's no other surveys like this that addresses these questions that clinicians don't ask women and often are overlooked because most surveys are unisex and clinicians only, generate ideas and a hypothesis from unisex data. So we're planning to launch this survey through 3 platforms, and we want to see the association between lifestyle and UPDR scores. So UPDR scores are basically scores that we give those with PD, and the higher the UPDR score is, the more severe your PD is, so we can track progression throughout time. And we're planning to launch this through Cleveland Clinic, PD Gene, and the Fox Insight, which are two biggest nonprofit organizations that collect genotyping and clinical data of PD patients in the community. And we're going to create prediction models looking at risk, severity, and progression. We also want to see if genetics can better predict the risk and progression of PD. And to do this, we're going to use the cohorts from PD Gene and Fox Insight and also actively recruit at CCF and genotype. And what we want to do is compare these models. Does just clinical and lifestyle factors help predict for women with PD the risk and severity? Or does it just the genetics? Or maybe by combining both these 3 factors, would that be the best prediction model? And with that, I'd like to thank my lab and all our collaborators. And with that, I'd love to take any questions.
Thank you so much, Shilpa. A little clap. Thank you so much for your presentation. Again, if anyone has any questions, feel free to add them into the chat, or feel free to unmute yourself and also ask a question. I want to say another giant data set that is going to be having to look through. I was wondering about some of, kind of going off of Jodie's presentation, some of the demographic information that you're taking through your survey as well to see, and are you going to be looking at all those differences as well with your data?
That's a great question. So the biggest problem is that, like Jodie said, a lot of our data and research comes from Caucasians. And so our lab actually has a consortium of Latinos from 35 institutions from the South America and the Caribbean. So we have this, as you saw in the survey, in Spanish. So we're hoping to collect Latinas, and we're hoping, because Cleveland Clinic is mostly Caucasian, we're hoping that through Fox Insight we can have a better demographic, but I would love to see more of that because we see that different demographics have different diseases more prevalent. So yeah, hopefully we'll have a big response like Jodie does. We're still actively waiting for IRB.
Good, sounds good, sounds good. Does anyone else have any questions? And another thing we can do is, if you do have a question, you can't think of it right now and you think of it later, feel free to also write them down in the chat. We can chat about them at the end of the presentations as well. That'll be great. All right, well, I think we will move to the next presenter now. Thank you again so much, Shilpa, for your work. Huge data set that you're going to have to work through. That's awesome. Thank you so much. So we will move next to Victoria Bernaud. Victoria is currently a 5th-year doctoral student in the Behavioral Neuroscience and Comparative Psychology PhD program in the Department of Psychology at Arizona State University, working in Dr. Heather Beaumont Nelson's Behavioral Neuroscience of Memory and Aging Laboratory. Her research uses preclinical models to explore the effects that surgical and hormonal alterations to the female reproductive system have on neurobiological and cognitive functioning throughout aging. She received a master's degree in psychology in spring 2020 and is also an ASU undergraduate alumna class of 2017 with concurrent Bachelor of Science degrees in chemistry and psychology. Awesome. Today, Victoria will be giving a presentation titled "The Cognitive Effects of highly selective progestin-segesterone acetate in a rat model of surgical menopause. Oh, awesome. So thank you so much, and take it away.
With that, my Wi-Fi just cut out, so I'm going to apologize in advance. If I break up midway through this presentation, I'm happy to answer any more context-related questions at the end of my talk. Thank you so much, Travis, for that introduction. Yeah, so I already had my introduction. Let's go ahead and get into it. So I'm going to talk very broadly just about menopause and hormone therapy as it relates to this talk before we jump into some data. So first, to define menopause, menopause is retrospectively diagnosed after 1 year of amenorrhea or missed period and occurs at an average age of 52 years. And in menopause, women can experience vasomotor symptoms as well as sleep disturbances and memory and mood disturbances, which are going to be critical to this talk. To alleviate some of these indications, women can take hormone therapy. And I have a figure just for you here showing the relative hormone levels across this menopause life stage. So going from premenopause into postmenopausal life, we can generally see that there's a decline in 17-beta estradiol, and there are complement increases in LH and FSH, for example. Now to get a little bit more into hormone therapy typically contains an estrogen as well as a progestogen. And here progestogen is meaning progesterone as well as synthetic progestins. Now there's clinical work as well as prior preclinical work that models hormone therapy that has found differing effects of estrogens and progestogens on the brain and cognition. And so to jump into some of that preclinical work now, generally, it's been shown that estrogens can enhance spatial learning and memory performance, but this is dependent upon things such as menopause model, dose, regimen, age, and other critical variables, to not really dive too deep into that. Progestogens, however, have been shown to be largely detrimental to spatial learning and memory processes. And I will note that some have been shown to be beneficial when given alone, but have been demonstrated by our lab in particular to be impairing with an estrogen. They impair cognition. So you can see I have a figure for you here of an example progestin, medroxyprogesterone acetate, or MPA. And what we found in our laboratory is that MPA administration resulted in cognitive deficits relative to control animals on a learning and memory task. Now, these findings contrast with the literature showing that progestogens can have neurogenic or even neuroprotective effects within the hippocampus. And I have this figure for you here on the right showing that a progestin called nesterone or segesterone acetate that I'll be talking about for the duration of this talk was shown to result in an increase in BRDU cell ratio relative to vehicle controls within the hippocampus. So we have some interesting findings when we look at spatial learning and memory processes that seem to contrast maybe some of the neuroprotective effects that have been observed within the hippocampus. This gets to the current experiment. So I evaluated cognitive outcomes associated with the administration of the progestin-segesterone acetate, or nesterone, as I showed you on the last slide. Segesterone acetate was selected because it has 100 times the progestational activity compared to progesterone. So I wanted to understand, how does something that has a high level of progestational activity impact cognitive outcomes? So we administered a low or high dose of this segesterone acetate, or MPA, to female 12-month-old rats that had undergone ovariectomy, which is removal of the ovaries, or sham surgery. And you can see I have the treatment groups for you on the bottom left. Now, after 5 weeks of this administration, we began a behavioral battery evaluating learning and memory outcomes. Specifically, I'm going to be talking about the water radial arm maze, or the RAM. We also evaluated anxiety-like behavioral outcomes. And so I'll also be discussing our results from the open field test, or OFT. First, to jump into these learning and memory outcomes from the water radial arm maze, we're going to be looking at performance in the acquisition or learning phase of the task. And what we found was that for one particular type of working memory error, it's a working memory correct error, there was a trial by treatment interaction between animals that received vehicle and animals that received the low dose of SGA. And when we looked at that highest working memory load trial, that would be trial 4, we found a significant effect where animals that received the low dose of SGA made significantly more errors than vehicle-treated animals. We also saw a similar pattern of effects for our other working memory error type that we evaluate, working memory incorrect errors. And so we again saw at that highest working memory load deficits with administration of SGA. And so the take home here is that there were working memory impairments that were observed during learning in animals treated with this low dose of SGA. Now to get to the anxiety-like behavioral outcomes, we're looking at outcomes in the open field. And just to give you a little bit of context, what we're specifically evaluating is small center time. And what we would expect is that animals that have a higher level anxiety are actually going to spend a smaller amount of time in that small center and spend a greater amount of time in the corners of the open field. And so what we find is that treatment with SGA at either the low or high dose reverses the anxiogenic effects of OVEX in the open field. So we see some reversal of anxiety-like behavior with treatment of SGA. Now, just to quickly jump into some future directions, we could evaluate the cognitive effects of SGA treatment in other menopause models, namely the VCD model. VCD, or 4-vinyl cyclohexene diapoxide, allows for an ovary intact model of menopause. VCD results in follicular depletion and also changes in ovarian hormone levels that more closely map onto the menopause transition. And I have a figure for you here on the right that is just demonstrating the effects of VCD on the ovaries. So you can see the leftmost side of the photo is actually a photo of an ovary, a rat ovary. And you can see that there are different cell types that are identified within those ovaries. And when we look to the right side of the figure, what we see is an ovary that was treated with VCD, and it is follicle deplete. We can see that there's not those same follicles that have been identified as in the left side of that panel photo. Some other things that we could evaluate would be the cognitive effects of SGA when paired with an estrogen. And this would more closely model combination hormone therapy, which many women do take. And we could also evaluate putative changes in other dimensions of cognition and behavior that extend outside of learning and memory outcomes, such as things like attention or cognitive flexibility. We could further dive into mood-related outcomes. There's a lot up to evaluate here. So with that, I just want to thank the Monty-Nelson Lab, my lab, my program, and our funding. And I will take any questions. Feel free to email me and also, follow me on Twitter.
Thank you very much, Victoria. Thank you. Awesome. Perfection. Awesome work. Does anyone have any questions for Victoria?
I can ask a question.
Oh, yes.
Yeah.
Great talk. So I was just wondering, for your behavioral testing, how many rats did you use?
Oh, sure. So they're going to be ends of 9 or 10 in each of those groups.
Okay. Yeah. And then do you plan to do any more severe anxiety kind of tests, like the radial arm, just like the high end, I forgot what it's called, the cross one?
Oh, elevated plus, for example.
Yes. Yeah.
Okay, yeah. Our lab typically looks at learning and memory related outcomes, and we've kind of since expanded to look at maybe affective like or mood related outcomes. Of that in my current, did I just freeze? I apologize if I just froze. I did. Yeah, oh, it just told me my connection was unstable. Sorry, let me start over.
You're back. Yeah.
Elevated plus is a great idea. We've just started to kind of delve into these affective like or mood behavioral evaluations. We've more, I think, kind of focused on learning and memory related outcomes. That's what our main interest is. I haven't done that for this project, but certainly we're interested in kind of more broadly looking at other cognitive and mood outcomes associated with critical variables surrounding menopause. Yeah, that's a great idea.
Thank you so much. Thank you. And yes, thank you for coming, Heather. I see your message down there. No problem.
I see you. Nice job, everybody. It was great to join you.
Awesome. Thanks for coming.
Have a good day.
See you later. Awesome. And a question also from me, for you, Victoria. I'm wondering if you're looking at different types of mechanisms of how SGA is kind of working. I'm wondering if you're looking at other things such as like corticosterone in these animals to see how it might be influencing that response and how that might be changing like gene expression or things of the sort.
Yeah, that's a fabulous question. Yes, so I will say these cognitive outcomes did surprise me. I thought that there would be maybe a little bit more of a direct relationship between progesterone receptor activity and beneficial cognitive outcomes. So I did evaluate another potential mechanism. I looked at, I did Western blots for GAD 65 and 67, so looking at any potential GABAergic modulation, and I did not find any significant effects for dorsal hippocampus, frontal cortex, and entorhinal cortex. I will say there were a few interesting correlations that I have forgotten the direction of, but that is to say that there were some interesting relationships there, but I don't think that that's necessarily the mechanism by which we're seeing these effects. I did not look at CORT. I think that would be really interesting, particularly given the open field results that we do have, but that's not something I've looked into.
Awesome, sounds good.
Thanks.
All right, well, thank you so much again. If no more questions, then we will move to our next presentation. And yes, we'll give another round of electronic claps for Victoria for sure. Thank you so much. All right, so we are moving to our next presenter, which might be our final presenter of today, we will see. And that is going to be, from Frankie Uy. Frankie is a graduate of UBC's Bachelor of Science in Behavioral Neuroscience program. In summer 2021, she was thrilled to work as a research assistant at Lisa Galia's Laboratory of Behavioral Neuroendocrinology, where she became interested in sex and age differences of stress in cognition and immunity. She is currently completing her Master of Management degree with UBC's Sauder School of Business. Though through science and business, Frankie aspires to improve scientific literacy, particularly around topics surrounding the neurobiological basis of depression and chronic stress. And today, Frankie will be giving a presentation titled Sex Differences in the Role of Frontal Cortex Activity in Negative Cognitive Bias from Adulthood to Middle Age. So thank you, Frankie, and go right ahead.
Thank you so much for the introduction. Well, hi, I'm Frankie. Well, yeah, you did my introduction already, so we can get into it. I just want to thank you, Travis, for giving me the opportunity to help you with your research throughout the summer, as well as Dr. Lisa Galia and the rest of the laboratory for their support throughout the summer. Okay, so cognitive bias is a subconscious systematic error of thinking, while negative cognitive bias is a tendency to interpret ambiguous situations negatively. So this is seen in depressed individuals. For example, if someone said, well, I've certainly never tasted chicken like this before, someone who is depressed will interpret the statement as negatively compared to people who do not have depression. Negative cognitive bias is a main symptom of depressive disorder. So for this presentation, things you need to know about depression include females report greater levels of depression and experience more severe cognitive symptoms compared to males. Middle-aged individuals report more severe depressive symptoms compared to younger individuals. And that depression is associated with altered functional activity in brain regions such as the prefrontal cortex and hippocampus and even nucleus accumbens. So despite this research, there is limited out there that examines the relationship between the sex and age differences and frontal cortex activity, functional connectivity in these brain regions, and how it plays in response to negative cognitive bias. So our hypothesis is, or was, there will be age-specific differences in frontal cortex activity and functional connectivity in response to negative cognitive bias. So the experiment included groups that were separated based on sex and age. So there were our male and female groups and young adult and middle age groups. And within that, they were further divided into two conditions: a shock period test group and a home cage control group. So for the shock period group, they're placed in a environment and administered a shock. And this was done every other day until testing day, when the subjects were placed in an ambiguous context and freezing behavior was measured. And if the subjects displayed higher freezing behavior, that would be indicative of negative cognitive bias. And if they had low freezing behavior, that would indicate positive cognitive bias. After that, CFOS immunoreactivity was measured. And this was chosen, CFOS was chosen because it reaches peak expression 90 minutes after being exposed to that event, with that event being the ambiguous context. And being able to look at the CFOS allowed us to look at neural activity and functional connectivity in frontal cortex subregions. So why the frontal cortex? Well, research indicates that the frontal cortex functions may play a role in negative cognitive bias. So subregions include the cingulate gyrus, which associates actions with emotions like fear, the prelimbic cortex, which plays a role in adapting to shifts in circumstances or environment or attention, and the infralimbic cortex, which inhibits emotional responses and suppresses fear. Okay, so on to our results. Okay, so these results show the CFOS density within each subregion of the frontal cortex, and a higher density, or the taller bar, would indicate more neuronal activity, and the stars above them would indicate that the data is significant. So we see that all groups, despite sex or age, showed more freezing and activation in brain regions compared to the home cage controls. And again, the rats that displayed higher freezing behavior displayed more, or would be associated with more negative cognitive bias. So it implies that increased neural activity in frontal regions plays a part in cognitive bias. Besides that, functional connectivity was also looked at. And so this is a heat map that shows the functional connectivity between brain regions. And for those who may not know, functional connectivity is a way to correlate the brain regions together to kind of see what operates together and what doesn't. So on the bottom, like the x-axis, you can see the subregions of the frontal cortex. And on the vertical y-axis, you can see the other brain regions that this is correlated with. And here the red would indicate more positive functional connectivity, meaning they interact together quite a bit, and blue indicating negative functional connectivity. Data here was only collected from rats that demonstrated negative cognitive bias only. So it allows us to kind of compare between the groups. So in these groups, you can see that in the females, it's more red overall. So it indicates a more like positive functional connectivity. And it means that like these brain regions interact more during ambiguous situations, or when placed in ambiguous situations. Of notable mention are the ventral hippocampus and the nucleus accumbens. Across young adults and middle age, they are still quite red. So they indicate more positive functional connectivity between the frontal cortex regions and these areas. On the flip side, the males are more blue. So on the left, we see the young adult males have strong functional connectivity between the ventral hippocampus, dorsal hippocampus, and amygdala. And in middle age, there is still some blue, but you can see that the blue isn't as dark. So it means that the males did lose some of this functional connectivity with age. So what are the key takeaways? Sex and age differences do exist, as you can see. So in the heat map I have here in the top one, we saw that males lose functional connectivity from young adulthood to middle age. And overall, females show more positive connectivity, especially in the frontal cortex and when it's correlated with the nucleus accumbens and ventral hippocampus. And that brings us to the end of my presentation. Thank you very much. I'm happy to answer any questions.
Awesome. Thank you so much, Frankie. Thank you.
Oh, thank you
so much. Thank you so much. Does anyone have any questions for Frankie? Stop me from asking a million. Thank you, Victoria.
Okay. Hi, Victoria.
Sure. Yeah, great job.
Thank you.
So I'm really interested in, I love that you looked at the effects of age as well as sex. I think that's really important to understand those kind of separately. But I think it's interesting, particularly in the context of female aging, to talk about changes in hormone levels with aging. So have you considered maybe digging in a little bit to looking at potential modulation of estrogens or progestogens and how that might alter this negative cognitive bias or this heavier, it seems that there's a greater relationship with aging for females towards a negative cognitive bias. Is that potentially, you think, modulated by ovarian hormones?
It could be. I'd say since I did this over the summer, I didn't have the opportunity to look at those ovarian hormones, but Travis was telling me how they were going to, in a future study, they're going to look at not necessarily ovarian hormones, but if I'm correct, it's other neurotransmitters such as like GABA and serotonin, and just seeing like how, like looking at the functional connectivity, seeing how those different levels come into play based on those correlations. So that would be really cool to look at, though. I think it would be really valuable as well.
Awesome, thank you. Yeah, I think that the next steps looking at like cholinergic or GABAergic functioning, it sounds wonderful as well.
Yes,
thank
definitely.
you.
And I'll add to that answer as well for you, Victoria, too, because we do have the data. Well, we have the data so far for collected lavage slides for these animals to see where they were at in their estrous cycle for the females. And at the moment, we were finding kind of no effect on negative cognitive bias with that, but we're going to look again and double check and make sure in the coming week to see that we didn't miss anything. We also collected blood samples so we can look at things such as estradiol versus progesterone. And I'm interested from what we've seen from the slides into actually progesterone, looking at that later on. So we will see what we find. I'll let you know.
Sounds good. Thank you.
Awesome. And any other questions too for Frankie? Awesome. I have one for you, Frankie. Yeah, no problem. Just want to know, because you talked about earlier in your presentation about the different functions of those regions of the frontal cortex. You don't have to talk about what each of them do, but I'm just wondering, seeing that there's more positive connectivity with those regions in the females, especially young adults, compared to the males, what do you think that means for their display of negative cognitive bias?
I was reading a little bit about this, and I was reading how like the ventral hippocampus is involved in stress responses and emotional behaviors, and like the nucleus accumbens is involved with reward. So I was kind of thinking that, your question is, what was your question?
Yeah. No, I was just wondering what your findings mean for their display of negative cognitive bias, the sex difference in display of negative cognitive bias. What do you think it means?
Yeah, I was thinking that like if the ventral hippocampus and like the nucleus accumbens, like the more that they send the projections to the frontal cortex or vice versa, the increase in activity, I think it would kind of link, I was thinking it could kind of link to how females report more like severe cognitive symptoms in depression, or report that, or even more effective depressive symptoms compared to males, which would be really, really cool, especially if they're involved. That's my interpretation of it.
Awesome. Thank you so much. Thank you. And yes. Well,
do you mind if I ask one more?
Oh yes, for sure,
question?
Victoria.
Do we have time? Do we have time to ask one more question?
Yes, we do.
So your question, Travis, sparked this question, which is, you had a group that underwent this kind of like repeated shock paradigm and then the novel context. Did you find a difference in incidence of the number of males and females that went into those like negative and positive bias categories? Because I think that's, if there is, that's interesting as well, right? Maybe if females are more likely to fall into that negative bias category in this paradigm than males, or vice versa.
To my understanding, I think the data was collected because we looked at CFOS right after, like 90 minutes after, we weren't able to measure like how long the freezing behavior was, or like even put them in the ambiguous context multiple times. I'm not sure if like the length of the freezing behavior was looked at. But I think that knowing that would also be able to elucidate the behavioral differences between males and females.
Yeah, because yes, your project, Frankie, was based on just looking at the brains of those animals that went through the procedure. But yeah, I'll tell you, Victoria, the young adults, males and females, they are both negative. We did in a bunch of different age groups. They both have negative cognitive bias, and it's similar in negativity. Whereas in middle age, actually, it's the males that are more negative than the females. And the females get to become more positive in the middle age group compared to the males, which is the opposite of what we were predicting before the study began. But yeah, that's what we got in terms of sex and age differences.
Interesting. Well, I'm excited to see more about this. That's wonderful work.
Yes, awesome. Thank you, Frankie, for helping me complete it. All right. Well, yes. Well, before we go, and before I start showing the ending slide, I am going to put those Indigenous charities I talked about before into links to those into the chat for everyone before I forget. Also a link to the National Day of Truth and Reconciliation. More information about that will be the third link that I'm putting down into the chat. But yes, let me share my screen. Ending presentation. But yes, thank you all again for your presentations. This was amazing. I always love the training presentation series because I love just hearing from other trainees. And also when it comes to questions too, I love hearing questions from other trainees. And I feel like this is a very kind of safe space to just chat together about what the research is that we're doing, and at all levels too. So thank you all for your wonderful presentations, and thank you all in the chat for coming and asking questions and things. That's awesome. This will be recorded and it will be uploaded later on, so you can check it out again to check out the research that everyone's doing once again.